Hi everyone, my name is Alexis Uria. Today is May 2nd, 2022, and I'm going to be sharing my Week 11 Digestive System portfolio. I did my PowerPoint on the short and long reflexes of the enteric nervous system. So the enteric nervous system is also coined the little brain. It is independent of control of the central nervous system. However, it does share several features with the central nervous system. They both have intrinsic neurons, neurotransmitters and neuromodulators, glial cells, diffusion barriers, and an integrating center. The ENS is known to control motility, growth, and secretion of the digestive tract. Starting with the short reflexes in the ENS, these are going to be the reflexes that originate and integrate within the ENS without external input. There's two main plexuses. The first is the submucosal plexus, which is going to control the secretion by GI epithelial cells. The myenteric plexus neurons influence motility of the GI tract. Local stimuli, such as dissension in the presence of food osmolarity acid, triggers two different paths. The first path starts with sensory receptors and neurons in the ENS. These are then sending input into the interneurons, which stimulate the enteric neurons and then control smooth muscles and exocrine cells and secretory cells, which are then acted on by GI peptides and cause several different effects. The first is changes in GI motility. The second is the release of bile and pancreatic secretions. And the third is enzyme, acid, and bicarbonate synthesis and release. The second pathway directly stimulates the secretory cells of the stomach and intestine, which then activates GI peptides and acts on the smooth muscles and exocrine cells to produce the above effects. So GI peptides excite or inhibit motility and secretion. They act as hormones or paracrine signals, and the paracrine peptides are secreted to the lumen and combine with receptors on the apical membrane or directly into the extracellular fluid and act on neighboring cells. So here you can see that the red arrows are indicating short reflexes in the enteric nervous system. Moving on to long reflexes, these are going to be the reflexes that integrate in the central nervous system. Long reflexes can originate from within the GI tract, but they can also originate outside of the GI tract, and these are known as the cephalic reflexes because they begin in the central nervous system. So feed-forward reflexes, the stimuli include sight, smell, sound, and thoughts of food, which then prepare the digestive system for food the brain is anticipating. Emotional reflexes can also stimulate this, and GI responses to emotions cause traveler's constipation, the feeling of butterflies in the stomach, and psychologically induced vomiting and diarrhea. During long reflexes, the smooth muscles and the glands of the GI tract are under autonomic control. This includes both sympathetic and parasympathetic divisions. The parasympathetic division is excitatory and enhances GI function, which is why it's known as rest and digest. However, the sympathetic division is inhibitory and inhibits GI functions. So the long reflexes are going to be reflected by gray arrows here in this diagram, or blue arrows, which are going to be seen in the next slide. Path 1 begins with cephalic phase of digestion and stimulates sensory receptors, which then stimulate the cephalic brain, which triggers sympathetic and parasympathetic neurons and stimulates the enteric neurons. This then stimulates smooth muscles and endocrine cells and secretory cells, which causes, in the first green box that you see on the right-hand side, changes in GI motility, release of bile and pancreatic secretions, and enzyme, acid, and bicarbonate synthesis and release. The second path of long reflexes begins with distension and triggers sensory receptors and neurons within the enteric nervous system, which then stimulates the cephalic brain and then activates the sympathetic and parasympathetic neurons. This directly stimulates enteric neurons, which affects the secretory cells of the stomach and small intestine. The secretory cells then activate GI peptides via short reflexes. The third and last path that we'll look at here begins when GI peptides act on the brain and also act on the endocrine pancreas. So the brain directly stimulates feelings of hunger and satiety, and the endocrine pancreas increases insulin and decreases glucagon. So to review the short and long reflexes of the ENS, I have mapped out all the pathways. The arrows are going to reflect both enteric nervous system pathways and pathways outside of the enteric nervous system. However, within the enteric nervous system, we went over the short and long reflexes. The short reflexes should be represented in red, and the long reflexes should be represented in blue. So you could take your time to go through this pathway and color in the arrows which you think would be short or long reflexes. And the answer key is going to be found on the next slide. So here, the short reflexes are in red, and the long reflexes are in blue, which appear a little bit darker here. But just to review, here are the stimuluses in green, which enter into either the long reflex, which goes into the integrating center, which then enters the enteric nervous system, or stimulus that directly activates the enteric nervous system. And on the boxes on the right-hand side, these are the tissue responses, which emulate the responses of the GI tract.